favorite part. So yeah, second seminar of spring semester. It's my great pleasure to introduce our seminar speaker, Dr. Kerry Nickols, who's currently a postdoc at Hopkins Marine Station. And she's a postdoc with the Marine Life Observatory there, and she's been doing a lot of really interesting things. Both Erica and I had introduced Kerry, or had invited Kerry to come speak this semester, so I think that was a good sign. Multiple different labs. I wanted Kerry to come talk to us about some of the work that she's been doing, really looking at biophysical processes. So Kerry got her undergraduate degree from Berkeley, and after she graduated, then she spent some time as a technician working on a variety of projects. She worked for Dan Reed for his SONGS project, working on the artificial reefs down off Carlsbad. And she did some work in the Bahamas with some of my colleagues and one of my best friends. Jamil was a graduate student there at the time. And so I first got to know Kerry 10, 12 years ago now, the first time that she came to the Western Society of Naturalists Conference. And since then, our colleagues and friend groups have really just overlapped, and so we've gotten to know each other really well socially. And haven't had a chance to work together yet, research-wise, but yes, we're going to make that happen soon. So while she was, so after some of those technician positions, she decided that she was really interested in looking at the effects of sort of flow in the nearshore environment and how that might affect dispersal and some of the ecological processes. And so she went and got her PhD at UC Davis, working with Brian Gaylord and John Largier. And that's some of the work she'll be talking to us about today in the seminar, really looking at what's happening in the near shore, sort of that, the closest kilometer to shore, and how processes of flow and things are being affected. And I think that's what's really great is that Kerry started as an ecologist and really got interested in learning some of these tools in physical oceanography to link ecology and oceanography. And so she's one of the rare people that's doing that. And the rare people that's really working in that near shore, that coastal boundary layer, which is a place that most oceanographers kind of ignore. So after Kerry got her PhD, she stayed on for a while as a postdoc at Davis and started working more on some models that were looking at population dynamics of fishes and the response to marine protected areas, and trying to link some of these models to empirical data instead of the models being very sort of abstract, trying to see if you can make ecological models that really predict the same patterns and the same densities and things that you would see from monitoring data inside of MPAs. After she finished that up, now she came to Hopkins, and she's been here for, has it been a year now? Working as a postdoc there out of Mark Denny's lab, and she's been doing a lot of interesting things, trying to look at how flow might affect the dispersal and the movement of zooplankton and other larvae through the kelp bed, which is really cool. She also just interviewed for the marine conservation job at CSU Monterey Bay, so hopefully we'll keep Kerry in Monterey for the future. So with that, Kerry.
Yeah, thanks, Scott. It's nice to also get an introduction that's so personal to show everybody just what it means to work in this field and to be able to make personal connections with your colleagues sort of throughout and opening up new areas of collaboration. And so it's been nice now being here in Monterey Bay and seeing Scott in this position. So because I was invited by both Scott and Erica, I decided to sort of talk about everything that I do because there was interest in both the the biophysical stuff and also in the population modeling work. So I sort of feel like the umbrella behind my research program is really looking at empirical observations and monitoring data, and then trying to move beyond that to go to a more predictive sense using modeling approaches. So I'm going to tell you about some of the work I did during my PhD, some of the work I did at my postdoc at UC Davis, and then at the very end I'm going to give you sort of a hint of the stuff that I'm doing now at Hopkins. And I think Monterey Bay is a really good example of why we need sort of this interdisciplinary framework. So I am very interdisciplinary. I have interests in ecology and in oceanography. And all of us know, Monterey Bay is situated within the California current. There's some really fascinating physical dynamics that are occurring, and the processes of upwelling and relaxation and how that affects this really diverse ecosystem. And the questions really that we have left to answer, and especially some of these challenging environmental questions about climate change really are going to require interdisciplinary research. And a lot of seminal work has been done right here within the bay. For instance, work on population connectivity, which was really started by Roughgarden and colleagues in trying to explore what we know about circulation patterns and how that influences how larvae are dispersed in the coastal oceans and what that means for the connections between populations along a shoreline. Really great work done on marine protected areas. As you all know, one of the most ambitious efforts at marine protection occurring right here in the state of California, particularly within the Central Coast MPAs. And then, of course, the kelp forests in and of themselves are really unique, fantastic habitat, and some really early work, being done in Monterey, for instance, in Monterey Bay. And so I'm going to tell you about these 3 different areas that I work on and hope to, advance our understanding of population connectivity in the coastal ocean, of monitoring and adaptive management of MPAs, and then this idea of kelp forests in a changing environment. So starting with the population connectivity topic, so for most marine organisms, the adult phase, so this picture is taken from right in front of the Bodega Marine Laboratory looking out towards the ocean. And most work there, ecological work is done, sort of right here on the shoreline. And if we're studying these crabs, for instance, that isn't the greatest dispersive stage of these organisms, and that really occurs in the larval stage. So we have larvae being released into the water column, being transported in currents, and one of the really interesting things about larval dispersal is that most organisms have a pre-competent and a competent stage. And so during that pre-competent stage, they're developmentally incapable of settling. So they have an obligate period of time when they're in the water column, and then they reach this competency, then they become competent to settle, and they can, settle right away or when they find suitable habitat. But there is this phase where they have to be in the water column. And so if we think about population connectivity and how populations are connected on a shoreline, we really need to think about what's going on in the water column as these organisms are being dispersed. So here's, an example. Oh, sorry, that slide's from the job talk, clearly. CSUMB right up there. So if you're, this crab that's located in, near Pacific Grove, for instance, maybe your larvae go north towards Santa Cruz. Maybe your larvae go even farther towards Point Reyes. They might go south towards Big Sur, or they might be retained. They might move some distance offshore and return back to settle near the shoreline. And so there's a lot of different pathways that larvae can take, and that becomes really important when we think about the spatial arrangement, when we're doing spatial management and thinking about marine protected areas. One of the mandates of the Marine Life Protection Act is that the MPAs function as a network. And so if we're going to think about them in the context of a network, we really need to think about how they're connected not only to each other, but to non-reserve areas as well. So that's my main sort of interest in thinking about population connectivity, and that's coupled with this sort of paradox, which is that so much attention with marine larval dispersal has been in thinking about the huge dispersal potential for larvae because they're in the ocean, they're being transported on the currents, and so they may go all the way to Point Reyes, they may go hundreds of kilometers, and so there's been so much attention at how far larvae might be able to go. And so there is this paradigm that larvae are swept great distances offshore and advected in the alongshore component as well. And that's juxtaposed against a lot of recent evidence that larvae are actually staying quite close to the shore as well as their natal site. Higher than increased self-retention is an idea that's been really prominent over the last probably 5 to 10 years in marine ecology. And this is occurring across different species. So work by Rita Borges in Portugal really focusing on fish. Stephen Morgan really focusing on crustaceans. Shanks and Shearman, that paper also has to do with mussels. So it's across, and this is just a small sampling. There's also genetic evidence and evidence from microchemical tags that things really aren't going as far as we once used to think. Here's just an example from work by Stephen Morgan. So we have distance from shore on the x-axis, abundance of larval crustaceans on the y-axis, and across all of these different larval stages, they're seeing the highest abundances of larvae just within a few kilometers from shore. So So clearly there's something really important going on just within a few kilometers from shore. And some of these larvae have behaviors that might keep them close to shore, but this evidence of higher than expected self-retention is really across a variety of organisms with different behavioral capabilities. And so I was really interested in thinking about what's going on close to shore. Is there something going on close to shore in terms of the water circulation that is able to explain this higher than expected self-retention that we're seeing across organisms in the marine realm? And for a lot of work that's been done right at the shore, if we want to incorporate physical measurements, sometimes that closest piece of information is a couple kilometers offshore in deeper waters on the 30-meter isobath, is typically where we have a lot of oceanographic moorings. And so there hasn't been, even though it's right in our backyard, there hasn't been a whole lot of exploration in this area that's right close to shore that really might influence larval transport. And this is the area, of course, where larvae are presumably starting and ending their lives. So if they, are released from the shoreline, advected some amount offshore and some amount downstream, ultimately, if they want to settle in suitable habitat again, if they're coastal organisms, they're going to have to then return to the shore. And so, this is a pretty important region that hasn't received much attention. And a lot of larval dispersal models will use one value of velocity generally to explain this whole region. So if you think of some circulation models, some ROMS models, although they are getting much better and better at sort of filling in some of those spatial resolutions. But in general, all of this area would be collapsed into one spatial cell. Also with things like high frequency radar, so this is surface current information, also doesn't really include this whole nearshore area. So this is where I focused my efforts during my PhD, this region right close to shore that may have some really powerful implications for larval dispersal. And the main questions that I was trying to get at that I'm going to talk about today are, do nearshore currents follow a generalizable pattern within this area? And then can these currents influence nearshore larval transport in a predictable way? So if we have some sort of a general mathematical description for currents close to shore, what does that actually mean when we start thinking about larval transport? So the work that I did for this part was done at these 5 stations along the coast of California, where I measured velocity and cross-shore transects, and they ranged in bottom depth and offshore extent, just depending on the site-specific bathymetric slope at each of these sites. Most of these deployments were sort of opportunistic. Other people were putting out instruments, and so it was, one more dot in my transect. Here's just an example of one of those deployments that occurred just north of here. And so here's the shoreline over here, and I had these 5 instruments spaced out between the 5 and the 20-meter isobath. I use acoustic Doppler current profilers, pictured here, and they profile velocity throughout the entire water column by using a sonar technique and then working off of the backscatter that's of particles in the water. I did most of these deployments myself. I am a total field junkie, and this was kind of the only big field component of my PhD research. And it's pretty funny when you start graduate school and your advisor's like, sure, take like 3 $30,000 instruments and drive them down the coast. And I'm very grateful for having access to these instruments, because that isn't something that, that most people would have access to. So now I'm going to walk you through some results from this deployment. This is just for one of the sites, but the trends that I'm going to show are consistent across all of the sites. So here we have time on the x-axis and velocity on the y-axis. And I also have a schematic to sort of guide you through looking at these plots. So here is the shore, for instance, and when I talk about the along shore direction, that's going to be velocity that's parallel to the shore. And then cross shore is perpendicular to the shore. You can think of as offshore or onshore. So these transects were cross shore transects. And the velocity measurements up here, this is the along shore velocity in the solid line, and then cross shore velocity in the dashed line. And I'm looking at depth average current, so looking at velocity integrated over the water column. And first you can see that velocity is strongest in the offshore or in the along shore direction. It's an order of magnitude order of magnitude larger, and so that's the component that I'm just going to focus on in today's presentation. So if we move even closer to shore, so that black line corresponded to an instrument that was the farthest out in this transect. Now you can see with this next line, as we move inshore, that we're getting lower magnitudes as well as lower oscillations in velocity, and then again moving further and further close to shore. So this idea that alongshore velocity decreases near the shore, we see this at all of the sites. This is the time series, but in order to compare all of the sites together, what I did was I took week-long averages, thinking of a time scale that is relevant to larvae dispersing. So I'm going to show you the data for all of the sites, for all of the locations within each site. So we have distance from shore on the x-axis, and then alongshore velocity on the y-axis. So here's the site from Mohawk down by Santa Barbara, the data from Pajaro just north of here, Huntington Beach down in Southern California. This site had the broadest bathymetric slope, Hopkins, which had a pretty steep bathymetric slope, and then Terrace Point just by Santa Cruz. So for all of these sites, we see that velocity decreases as you go towards shore. And this idea that you have decreasing velocity towards shore, towards some sort of wall, decreasing velocity towards a wall, is a phenomenon that's actually been really well studied. It's most well studied in small scale boundary layers. And it's such a pervasive phenomenon. It even has a law named after it, which is the law of the wall. You know, in physics, there's laws. Ecology, we don't really have laws. So the law of the wall describes velocity as a function of distance from the wall. And then that's non-dimensionalized by the roughness of the wall. And then this U star parameter describes the velocity gradient, and then kappa is a constant. So if you non-dimensionalize velocity and distance from shore and plot them on a log scale, you get this universal relationship that velocity is related to distance from shore. It increases logarithmically as you move from that wall. So I wanted to see if this law of the wall logarithmic boundary layer effect, if that is at all analogous to what we see going out in the coast, while recognizing that the physics are completely different, but just trying to come up with this general description of velocity as a function of distance from shore. And so I plotted my data along with non-dimensionalized them and plotted them, and indeed they are all logarithmic. And we do see this general trend of velocity increasing logarithmically away from the shore, our wall in this case. This was a pretty surprising result because we have very different bathymetries, we have different topographies at each of these locations, and also I only showed you 1 week from each of these different sites, but of course I have a lot more data than that, and this pattern holds across time at these different sites. And if you're interested in that, I can talk to you about it afterwards. So this gives us this mathematical representation of water velocity. So these are velocity vectors. This is a schematic of this coastal boundary layer. So now we have this description of velocity changing as a function of distance from shore within this nearshore area, trying to fill in this region that's been left out of a lot of representations of transport in the ocean. And we can start looking at how this might affect descriptions of larval transport. I've also spent some time thinking about the mechanism that creates these logarithmic boundary layers. If you're interested in that, again, I can tell you about it, but I'm not going to go into it here. So for the research questions, do nearshore currents follow a generalizable pattern? Yes, they do. And now we're going to look if these currents can tell us something about nearshore larval transport that maybe can help inform this idea that we have higher than expected self-retention for a lot of species. And so the way that we're going to do this is to look at simulations of water movement in the ocean, and we're going to simulate larvae being released, and then we're going to look at who settles and where they settle. And the way that we look at that is by creating dispersal kernels, which are probability density functions. You can think of them like a histogram. So on the x-axis is a longshore position, and on the y-axis is the probability of settlement. So if we release particles or larvae from here, we might, under 2 different flow scenarios, get 2 different dispersal kernels, so distributions of settlers. And so for a release that had a short larval duration, we may see more settlement closer to the release site than for that that had a longer larval duration that was in the water column for longer. We can look at things like the mean dispersal distance, which in this case would be about 60 kilometers down coast, and we can look at this mean dispersal distance of this longer transported kernel here, and that means about at 145. So if we put a marine protected area at this place along shore, then a larva that had a shorter pelagic larval duration would end up settling within the marine protected area. But if that species, say, had a longer pelagic larval duration, represented by this dispersal kernel, maybe none of them would end up within the marine protected area. Conversely, if it was further down the coast, then this species here would, maybe seed the MPA more than this one down here. So that's sort of the theoretical framework for thinking about how transport might influence larval dispersal. There's another quantity that I'm really interested in, self-retention. And for here, I'm defining it as the fraction of released larvae that return within 10 kilometers of the release site. 10 kilometers, somewhat of an arbitrary number, but in some of the literature on the size and spacing of marine reserves, we think about sites maybe being within a sort of 10-kilometer area. In some circulation models, ROMS models, they define a site as a 10-kilometer width. And so for this situation here, we have 3% self-retention. For this situation here, less than 0.1% self-retention. And this also becomes important when we think about persistence of populations and if that population is able to maintain itself just receiving input from itself, and how much it may rely on input from an outside population contributing. And so if you want to be self-persistent, you need to have at least some amount of self-retention. So I constructed a model with a linear coastline with 750 consecutive 1-kilometer habitats. I modeled a shallow subtidal organism that spawns within the 5 to 10 meter isobath and settles within the 10 meter isobath. So you can think maybe algal spores or abalone, pick your favorite nearshore coastal organism with a somewhat shortish pelagic larval duration. I looked at pelagic larval durations between 3 and 30 days, and I did that by modeling equal pre-competency and competency windows. So again, returning to this idea that there is a window of time within which most larvae are unable to settle. They still have to continue developing. And then they reach a point where they are competent to settle. And in laboratory studies and in some field studies, there's indications that these windows tend to be equal. So that's how I modeled them for this component. And then I modeled them as passive particles because this really is sort of a null model of looking at the potential effects of the flow in absence of behavior. Now, of course, there could be behavior, which might, impact and influence larval transport, could lead to even higher amounts of retention if they're able to, maybe stay towards the bottom in the slower flows that we generally tend to see within the bottom boundary layer. So here's examples of dispersal kernels that result from these model simulations. This is for a pelagic larval duration of 5 days, and we have a longshore, or 15 days. We have a longshore distance on the x-axis and probability of settlement again on the y-axis, and this is just showing you for 2 different sites. And I'll note that this no coastal boundary layer case, this is by assuming that there is a constant velocity and also a constant mixing term that is the same throughout the spatial domain of the model. And this is sort of the business as usual larval transport sort of case. It's assuming that you take velocity from about the 30 meter isobath and then use that to predict larval transport. And so these huge peaks in settlement that you see in both of those cases have to do, for both of these sites, have to do with that pre-competency window. So larvae are being advected and mixed downstream and offshore. They reach the end of that competency window and they can settle, so you might get this huge peak in settlement. But then you also get these long tails, these long fat tails, and that has to do with them still being advected and mixed downstream and then finding nearshore habitat as they go. So if we add a coastal boundary layer into the model, we see that these dispersal distributions move towards the left, closer to the release site, but we still see these long, fat tails. So even with considering a coastal boundary layer in the model, you get higher settlement closer to the release site, but you can still have long-distance dispersal. So let's go back. So now what we're going to do is look, so this is just for 2 of the sites and for 2 different pelagic larval durations. So now we're going to show the data in a way that allow you to see the results across all of the pelagic larval durations and for all of the sites. So pelagic larval duration is on the x-axis, and then on the y-axis is the ratio of the mean dispersal distance with a coastal boundary layer to that without a coastal boundary layer. So if you're down here in this part of the plot, it means that the coastal boundary layer is important. And up here, the coastal boundary layer doesn't have a big effect on the predictions of mean dispersal distance. And so what you can see is that for all of these 5 sites and across all of the pelagic larval durations, there's at least some reduction in mean dispersal distance. However, we see the greatest amount of reduction for low pelagic larval durations, and then for these sites here that tend to have really broad bathymetric slopes. This site, Mohawk, that had a pretty steep bathymetric slope, and it also had really high velocities. We don't see, there's some reduction in mean dispersal distance, but not as much as for some of these other sites. Now we're going to look at self-retention. So here's pelagic larval duration on the x-axis, and then on the y-axis is the fraction of released larvae that are self-retained. So if you're at this level, that would mean that all of the larvae that were released were self-retained. And if you're down here, so I released 10,000 particles. So if you're down here, it would be that one of the larvae that was released is self-retained. The open bars are for the case of no coastal boundary layer in the model, and then the solid bars are with a coastal boundary layer in the model. I'm just showing you the results from 2 sites, but it's consistent across all of the sites. Without a coastal boundary layer in the model, we only get measurable amounts of self-retention for a couple of the sites, and not for very many different pelagic larval durations. But when we include a coastal boundary layer in the model, now we can see self-retention across a broader range of pelagic larval durations and for all of the sites. And self-retention increases up to 3 orders of magnitude. And so this is a pretty big result in thinking about this idea that there's higher than expected self-retention seen across a bunch of systems. And the coastal boundary layer provides a physical mechanism that at least partially explains those results. And this idea that larvae can stay close to shore and they can do it without even having any behavior. And it's important too to think that the coastal boundary layer can lead to both high retention, but you can still get those long tails. So this isn't shutting down any work saying that larvae do go far, because they absolutely can and do go far. But there's also a lot of them that stay close to shore. And if any of you are familiar with the dispersal literature in terrestrial systems and thinking about dispersal of seeds, in that system, a lot of attention was first put towards understanding dispersal right close to the parent plant. And then over time, people started looking at long-distance dispersal and thinking about how fluid structures associated with the canopy within a forest led to uplift and filled in this idea of long-distance dispersal. And then in that system, you see the same kind of kernels with this really high peak close to the parent plant, and then this really long, fat tail. And for a long time, I think we really thought that dispersal in the ocean didn't operate anything like that. And so the dispersal literature between terrestrial and marine systems has really been kept separate. But this is a physical mechanism that shows that we actually might have a lot of dispersal really close to the parent population while still having those long, fat tails. And if we don't include a coastal boundary layer in our thinking of how populations are connected, we may in fact be overestimating the level of connectivity between different populations. So on that note of thinking about how we can use empirical measurements and incorporating them in a modeling framework to try to understand how systems are really functioning, I'm going to move on to some work that I've done looking at monitoring and adaptive management of marine protected areas. So some example goals of marine protected areas are to protect diversity and the abundance of organisms. We want to sustain and conserve marine populations and also rebuild depleted populations in the case of fishing pressure. But no matter which goal is your favorite goal of why we should have marine protected areas, whether you're trying to save biodiversity or you're trying to rebuild a population, the bottom line is you have to have a persistent population. And we won't know if marine protected areas are working if we haven't considered expectations for responses, which require some knowledge of this idea of population persistence. There's two major effects of fishing on a population. So if we go from this unfished age structure, here we have age on the x-axis and then abundance of fish on the y-axis. When we fish that population, we end up with a truncated age structure with these fewer old fish, but we also get reduced overall abundance because we have fewer new fish coming in as well. So if we put in a marine protected area, in this case a marine reserve with no fishing, the recovery will depend on the time scale it takes to fill in these older ages, as well as the time scale of new reproduction, because I'm sure you all know that old big fish are not going to appear instantly. And so the group that I worked with and is still working, because of course that's what happens when you have a postdoc, it never quite ends. We use population models to try to understand what's happening to fish in marine protected areas. So we combine what we know about age and growth of fish, things like larval recruitment and fishing rate, into a mathematical population model of fish, and we try to look at how much change we should expect to see after an MPA goes in, as well as how fast that change might occur. And then we can compare our predictions to actual monitoring data. The monitoring data can help inform our model, and hopefully our model can inform how that monitoring data is collected and what we're actually getting out of that monitoring data. And this sort of process here of sort of going back and adjusting your models and incorporating monitoring data, is really mirroring, oh sorry, I'm jumping ahead of myself. So this whole thing, we're hoping, can help us look at what influences responses of fish populations to MPAs. And then this sort of cycle here of informing your approach with monitoring data is really what's supposed to happen with adaptive management. And adaptive management is really important when we're thinking about our marine protected areas in California. They're supposed to be managed adaptively. That's still sort of an open question what that means and how that's going to take place. But I hope you'll see that the work that we're doing in using population models to detect effects of marine reserves is something that should maybe be incorporated into adaptive management. So this work was done within the framework of the Central Coast MPAs, and today I'll just talk about some of the work that I've done looking at data from Point Lobos area. So Point Lobos used to be this reserve here in yellow, which has been no-take since 1973. Then as part of the MLPA process, that reserve was expanded to include the area here in red. And these circles that I've indicated throughout are areas where PSCO has been monitoring the subtidal kelp forest fish populations. And so they're collaborators on this project, Mark Carr and Dan Malone. So we've been working with this data set. And specifically, one site of interest is Monastery, which was previously outside of the old MPA, but now is encompassed within the expanded reserve as of part of the MLPA process. We are looking at a few different species, but today I'm just going to talk about blue rockfish, which is a nearshore kelp forest fish that's abundant in surveys with recreational and commercial take, and there's life history information specific to Monterey. So first we're going to look at what we might expect to see inside and outside a reserve through time. Here we have year on the x-axis, and on the y-axis is number of fish per sample. And this is greater than the fish length because we're really just thinking about those older, bigger fish that are available to fishing. And I've indicated here when the MLPA went in. And so if this is the trajectory or the data that we have for outside fish abundance, outside the MPA, then we might expect the inside abundance to be higher than that and persist through time. And when that new reserve went in and was expanded, Monastery, maybe abundance started to go up. There's also the effect of mean size potentially increasing as a function of MPAs going in. So now we have time on the x-axis, and on the y-axis is the mean fish length. Again, just looking at those fish that are available to fishing. So if this was the time series of abundance outside, then inside we probably expect that there's higher abundances of fish. When that MLPA was expanded, then we might start to see fish size increasing. So this is maybe some lofty expectations based on how marine protected areas might function. So here's what the data actually show from PSCO. So we have maybe higher abundances of fish outside of or inside of the MPA before the MLPA, but a lot of overlap with those error bars. And then the new reserve goes in. 2007 was just apparently a really bad year for blue rockfish, so we don't see very much blue rockfish inside or outside. And then mean size is kind of all over the place even before the MLPA went in. So why aren't blue rockfish more abundant within marine protected areas? Well, it could be that there was little fishing at the sites before MPAs went in. So if there wasn't a whole lot of fishing, then taking away that fishing isn't going to change much. It could be that variable recruitment is obscuring the effect of MPAs. So if you do any diving around here, you probably know that rockfish recruitment is notoriously variable. It could also be that big fish aren't resident in the kelp forest. So the big fish aren't in the kelp forest, so they're not being observed during these diver surveys. And these are not experiments that you can do. And so this is one reason that we can use modeling as an approach to start to answer some of these questions. And then also I should note that this idea of the little fishing, there's stock assessments of blue rockfish and for other species within this ecosystem, but those generally take place over a huge geographic area. So the stock assessment for blue rockfish is over the whole state of California, but here we're just trying to understand what's happening within this MPA. And so we really need to have some sort of local estimate of fishing mortality. And so the way that we did that is, again, to use population models to try to interpret this monitoring data. And to answer the how much fishing occurred question, we used a Bayesian approach where we fit the model to the data and try to estimate the local amount of fishing mortality that occurred. And this is the distribution of fishing mortality rates that results from thousands of these model iterations, and it has a mode of 0.3228, which is pretty consistent with levels of fishing predicted from the stock assessment. So we think that it's pretty safe to say that there was fishing going on before the MPA went in. So the next question is what role recruitment plays in obscuring the effect of marine protected areas. So we know that rockfish recruitment is really highly variable. Here's data from Jen Cassell and colleagues for these two different species complexes. If any of you were diving this year, you would have noticed that there were a ton of baby blue rockfish, but no baby kelp rockfish. So, maybe related to oceanographic variables, not super clear for the Central California region. So what we do is we incorporate the variability of recruitment into the model. And I'm going to show you some example trajectories. So this is taking that model of fish population change and running it under different scenarios and looking at the expectations through time. So we have years after MPA implementation on the x-axis, and then again, a fish abundance on the y-axis. So maybe for 10 years, this is with continued fishing, or reserve, again, with variable recruitment. Here's the output after looking at after MPA implementation out to 50 years. So we're going to step through a few different trajectories, but you'll see that there's overlap in outcomes with or without fishing, especially in the first 10 years. But of course, we don't just run a few trajectories, we run thousands of trajectories. And because there's a lot of variability, there's going to be a lot of overlap in outcomes and a range of variability in the model output. So what we do is we end up looking at the distributions of outcomes. So here's, again, years on the x-axis, number of fish on the y-axis, and then the solid lines are the median values, the median simulation output. And then these gray shadings are giving you the 25th and 75th percentiles. And then where they overlap is this dark gray area. So you can see that with or without fishing, and including variable recruitment, it becomes really difficult to tell apart the reserve or continued fishing outcomes. Now, one thing we could do is to say that, well, what if recruitment is just really amazing every year? If we have really great recruitment every year, then we get this much clearer separation of reserve versus non-reserve outcomes, but a huge overlap within the first 10 years after MPA implementation. Now you may wonder what's going on here with this huge increase, and that's because the 2007 abundances, which are the starting point for running these simulations forward, were really low. And so any years of really good recruitment are going to allow that population to increase a lot. I've also done the opposite, which is to have low recruitment every year. And what happens with that, especially because these abundances started out so low, and you saw through that time series that in 2007 there was that low point. So if we look at just really bad years of recruitment every year, then both lines are like down here and they don't change and they just stay the same. No difference between inside or outside, but there's also like no fish. So the next question is to start looking more carefully at these size distributions. And now we're going to answer the question of what if the big fish aren't there? So this is the expected size distribution from a fish population model. So we have like a lot of babies, and then with the reserve, we're seeing these older individuals, bigger individuals, and then with fishing, we get less big individuals. So reserves should lead to this filling in of the size distribution. So we can put up actual size distribution data from the monitoring efforts and see if we're seeing those big fish inside the reserve that match up with our expectations. And there's some big fish, but really not as many as we expect to see under reserve conditions. So there appears to be this absence of big fish inside the MPA kelp forest. And thankfully, these monitoring programs aren't occurring in a vacuum, and there's multiple monitoring programs going on. And so we began collaborating with Rick Starr and noticing this observation. So here we have size on the x-axis and abundance on the y-axis. And with the kelp forest data, they're limited by depth, and they're not seeing as big of individuals as they're seeing with the hook and line survey data that's taken place in deeper waters. And that really, we can't just look at what's going on in the kelp forest to understand a fish that's occupying habitat larger than just in the kelp forest. But knowing these two different size distributions that are coming out of these monitoring programs, we can fit this selectivity curve about observations of divers as they're taking on these surveys. So we can account in our model for the fact that divers aren't observing these big fish. So if we incorporate this piece of information into the model and then run our simulations forward again, just to show you, so here's what the expectations look like for that size distribution without incorporating that we're not seeing the bigger fish in the kelp forest. If we incorporate that in the model, then these curves get shifted down, and now this is the expected observation for the reserve, which matches quite well with what we're seeing in the data. So now then, if we take this information and we run those simulations forward, we find that within the kelp forest habitat for this particular species, we wouldn't expect to see a difference between continued fishing and within a reserve even out to 50 years. And this is really, a call for why we need multiple monitoring programs going on simultaneously, especially for these species that are occupying different habitats. Now, we're running these models with other species, and so, we should be able to get a clearer idea with some of these species that maybe aren't going under these ontogenetic migrations. So I hope I've convinced you that evaluating MPA performance really requires expectations. So these MPAs are going to be up for review. There was a meeting I'm sure some of you were at last year, really looking at, 5 years after the MPAs went in, where are we now? Are these things working? And for these fish that take a long time to reproduce, we can't evaluate that question within 5 years. And in the case of some of these species that are migrating to offshore waters, if we only looked at one piece of the monitoring data, what are you going to tell a manager about how to manage that species and whether or not these MPAs are working? So models help us develop expectations and interpret monitoring data, especially in these cases where we can't do the experiments. And really, models are a way to start looking at some of these ideas in a mechanistic way. And they can also help us inform the design of the monitoring program. So as the Ocean Science Trust is going to be funding new monitoring programs, and hopefully, work like this will point to, yes, we need to be monitoring in multiple habitats for these species that expand across these areas. So in the last few remaining minutes, I just want to give you a little sampling of some of the work that I'm doing within the kelp forest in front of Hopkins, because I think some of you will be really interested in it and may just want to make you aware of this data set that I'm collecting and see how it might help inform some of the work that's going on here. I don't think I have to tell you that kelp forests are a critical ecosystem. They're a foundation species. They provide habitat for really important species, large amounts of biodiversity, and they're extremely productive. And there's also a lot of really fascinating physical processes that are taking place within a kelp forest. They're embedded within my beautiful, fun coastal boundary layer. But we also see differences in flow within kelp forests, so we can get lower flows inside of the kelp forest. We can get acceleration around the edges of beds because water, if it's going to move slower in the kelp forest, then that water has to go somewhere, so it may be diverted around the edges. So a really interesting physical environment, but there hasn't been much work in thinking about how the physical dynamics associated with the kelp forest influence biological processes, and also how they might influence some of the biogeochemical processes that are occurring. And kelp forests, embedded within all these physical processes, also embedded within this changing environment. With global climate change, we're seeing changing temperatures, increased storms and sea level rise, and also ocean acidification, the other CO2 problem. And there's not a whole lot known out there about the biogeochemistry of the kelp forest environment and how the physical processes within the kelp forest are influencing that environment. And so this is something that I've sort of taken on. It's a collaboration with a graduate student up on the main campus of Stanford University, Dave Coeek, who is a student of Rob Dunbar's. And we're really trying to get a handle on the biogeochemistry of the kelp forest environment so that we can make informed predictions about what's going to happen to species within the kelp forest. Just to give you a brief review of some of the carbonate chemistry system parameters, DIC is the sum of inorganic carbon species. You can think of that as the amount of CO2. pH, the measure of acidity via hydrogen ions, and then omega aragonite, which is the saturation state of aragonite. So this is when it's below 1, it means that we start having dissolution of calcium carbonate structures. And as CO2 or DIC increases, then we see decreases in pH and saturation state. In kelp forests, it's kind of unclear what's going to happen with them, because although we have inputs of anthropogenic CO2, upwelling, and respiration that all lead to decreases in pH, increases in DIC, and then decreases in saturation state, it's a primary producer and it's extremely productive. And that's acting to increase the pH by taking up CO2, leading to decreases in the amount of carbon in the water column, as well as changes in the saturation state. And so predictions of how kelp forests are going to respond to ocean acidification is really unclear, because there's all these different processes going on, especially in a region like this that is such an active upwelling area. So the questions that we're trying to get at are, what is the natural scale of variability in biogeochemistry in the kelp forest? How do physical and biological processes play into that? And then how the system responds to episodic events. So again, this is ongoing work, and today I'm just going to give you a little bit taste about this first question. So I've been working in the kelp forest in front of Hopkins. We've been looking at these 6 stations. This is also part of work that I'm doing looking at differences in larval distributions as a function of flow environments within kelp forests. So I also have current meters inside and outside the kelp forest on both sides here. This brown is just showing the outline of the kelp forest because Google people decided to take this picture on a really glary day. And so we sample all 6 of these sites. We've been doing this since July. We go out every week and we take measurements 1 meter from the surface and 1 meter up from the bottom. And today I'm just going to show you data from these 2 sites on the protected side of the kelp bed, but inside and outside the kelp forest. So we go out, we take vertical profiles with a CTD to get at temperature and salinity and density. We also measure some other things, but the ones most relevant for the carbonate system are density and temperature. We use Niskin bottles to take those samples, and then we poison them with mercuric chloride to cease any biological activity that's going on. We measure DIC and we measure total alkalinity, and that's because to get at the carbonate system parameters, you have to measure at least two things, and DIC and total alkalinity are the parameters that the biogeochemists have the best handle on those measurements. In full disclosure, I am not a biogeochemist, which is why I'm collaborating with a biogeochemist. So if you have any nitty-gritty things you want to know about how these samples are analyzed, in my world, they go in a box and then they get driven up to campus, and then I never see them again, except to present my data. Our data. It's our data because it's a collaboration, of course. So we have time on the x-axis, depth on the y-axis. So you're going to end up with these surface samples are going to appear here when I finally unveil them. And then we have the deeper samples are going to appear here. We're looking at inside the kelp forest and outside the kelp forest. So we take samples of DIC and total alkalinity, but today I'm going to present then you magically calculate all the parameters, and we know the temperature and the salinity and the density from the CTD cast. That's the data set that I work the most closely with. And then you can calculate the amount of PCO2. So that's what I'm going to present today because it's something that you might be familiar with. And to put it in context, this blue arrow here is showing you the pre-industrial level. That cyan is around the current level. And then the 2100 best case scenario from the IPCC report shows that up there just below 600. And then off the charts at 950 is the 2100 worst case scenario from the IPCC report. So now let's look at data from the bottom. And then here is the data from the surface samples. And what you can see is that both inside the kelp forest and outside the kelp forest, we see the highest concentrations of PCO2 at the bottom. Makes sense because at the surface we're going to have the kelp canopy. It's going to be uptaking CO2. We also have upwelling occurring, bringing nutrient-rich, high CO2 water from the depths. And we also see that PCO2 is highest outside of the kelp forest. So again, not influenced by the kelp taking up the CO2, so that makes sense. And then, oh man, that little thingy didn't show up. So the other really interesting thing, which is really obvious from looking at these graphs, is this huge variability between the top and the bottom. So there's gradients greater than 300 microatmospheres between the surface and the bottom. Now, when some people do ocean acidification experiments, that might be the level of a treatment difference. Yet here in the kelp forest, in the natural environment, we're seeing those sorts of variations just within the water column. Here's looking at saturation state. I apologize for the yellow. It's probably difficult to see. Time on the x-axis, saturation state of aragonite on the y-axis. So we don't ever really get below 1. That's good. We do get some decreases in saturation state that are below 2, and some organisms have shown responses to saturation state at 2. So this is, something to think about. So here's also laying on, so I was just showing you the protected sites, but if we look at what's going on in the exposed part of the bed, you can see that we're actually seeing even lower saturation states. So there's some really interesting spatial variability just between the protected and the exposed parts of the kelp forest, and that's something that I'll really be looking into further with all the physical data that I have out, the current meter data, and what we can do is we can look at certain days where we have different conditions and look at the carbonate system environment that's associated with different advection conditions. I also have data on kelp density, so we can really start to tease apart what's going on and driving some of this variability that we've observed. You also may be saying, well, you're just going out once a week and you're sampling once a day, like one time within that day. There's a lot going on. There's a lot going on with the kelp and the process of photosynthesis and respiration that's occurring over a day. So in answer to that, we are going out seasonally, and we did this last week where we went out every day of the week, and then for one day we went to 2 of the sites once an hour for 24 hours. And so we'll be doing that seasonally to really try to get a higher resolution look at what's going on in the carbonate system environment. So again, some of the next steps, this high resolution sampling and then a detailed analysis of the physical and biological drivers behind this pattern. The idea being that eventually maybe we can come up with some predictive model about which conditions are going to give you a certain carbonate system environment. So today I've given you sort of a seemingly hodgepodge presentation of population connectivity, monitoring and adaptive management of MPAs, and kelp forests in a changing environment. But in my head, these all fit together quite nicely within this idea that, you know, we really need interdisciplinary collaborative research to answer the challenging questions that are left to answer, and these can be the most fun to answer, assuming you have, you know, a great team to work with and combine resources and knowledge. And that's really important when we think about this environment within Monterey Bay that's so dynamic and has a lot of different things going on. And it really takes looking at both the physics, the biology, and then also the human use component of that system. I have a lot of collaborators, obviously, because I do a lot of really interdisciplinary work, and I don't have the resources to own all the instruments that I use. So I just want to acknowledge collaborators from UC Davis, from UC Santa Cruz, from Hopkins, Rick Starr here at Moss Landing and Sea Grant, Will White at UNCW, who used to be in California. That's why he's the oddball at North Carolina. And then Kurt Storlotzy at USGS, one of my current collaborators that helped instrument the kelp forest that I had over the summer. And of course, thanking my past programs for funding. And then I do a ton of field work, and it takes a lot of people to get that done. And thank you for having me, and I'm happy to take questions.
So questions, I think you can either speak into the microphone or talk really loudly in that thing. Like that.
So sometime in the future, there's some agency that's going to decide whether MPAs are functioning as they got to do or not.
Yep.
And you talked about blue rockfish, but it's one of many, many things.
Yep.
So are there specific criteria that are actually, I mean, will it actually be possible to make a decision? It seems like it's just a matter of involvement.
Yeah, I agree with you that that's what it seems like. And I wasn't involved in the MLPA process when it went through. My collaborators were. I have been to some meetings. There was a recent meeting, I don't know if anybody went to, there were like 3 different sort of town hall-ish type meetings about the monitoring that Ocean Science Trust put on. So there was one in Pacific Grove, one in Santa Cruz, and then one like in Morro Bay or something. And you know, they're trying to gather ideas for how monitoring should proceed and what we should be looking at. And I raised the question during that meeting of, well, we need to be measuring whatever they're going to be evaluating the MPAs based on. And that was sort of like, oh, well, yeah, but we don't really know what that is yet. And it's just a really, you know, complicated Venn diagram of, you know, the commission, the Fishing Game Commission, and the Ocean Science Trust. And my take on it is just that they aren't going to make they're not going to ask, you know, hey, Kerry Nickols, we really think that your work with population modeling is great, and we'd like to, you know, incorporate you. They're not going to do that. I need to be, you know, as scientists, if you want to be active in the MLPA process, I really think it's up to us to keep asking those types of questions and to be sort of active in that community. But at this point, it seems like there are no recommendations yet of how they're going to be evaluated. Yeah.
I was wondering regarding the law of the wall and dispersal models. So you applied the law of the wall in kind of a horizontal plane, and I was wondering if there's been any work done looking at the buoyancy of different larvae, because I would assume that they're positioned, I mean, if the law of the wall works horizontally, it definitely works vertically.
Yeah.
And if the buoyancy of the larvae at different phases affects also their dispersal.
It definitely does. I think it's been most probably worked out for crustacean larvae. Those tend to be the most well studied. They're big-ish in size. And barnacle cyprids are pretty buoyant. That's when they are in their competent stage. The nauplii are less buoyant than the cyprids. And crustaceans do, some of them have pretty complicated behavior. So some of them are staying close to the bottom. If you stay close to the bottom, you're going to be in even slower flow, so that might, influence some of these ideas. Some of them also vertically migrate, and they're sort of taking advantage of currents going in different directions at different times of day and night. And that's something that, so all of that, data that I showed you, that was, a monumental amount of work, but that's just scratching the surface. And so sort of the next step that I'd like to take that work, which hopefully I would have, a master's student or somebody who would help me do something like that, would be to really look at what's going on at the different positions in the water column and thinking specifically about which behaviors different larvae are undergoing, and then, making a three-dimensional model with that variation.
I have a question about the coastal boundary layer.
Yeah.
So the law of the wall, right, assumes that the closer you get to the wall, the closer you basically go down
Yep.
to nothing.
Yeah.
But when you think about sort of waves moving ashore, right, in the
Yeah.
shore environment, water's moving ashore, it has to go somewhere. And I try to think about I'm going out surfing with the
Yeah.
waves coming in, right close to shore, the water's going to be affected pretty quickly
Yeah.
along the shore, and then they have to find a channel to get out. So
Yeah.
is there anyone thinking about how are those processes, I guess, important to thinking about induction of larvae
Yeah.
that are born on shore?
So I cheated, and I put all my instruments outside of the surf zone. The surf zone is crazy complicated. There's work by Jamie McMahon at the Naval Postgraduate School, who's done a lot of work within the surf zone right here in Monterey Bay, and some of his work indicates that actually, it seems like you're being infected pretty far down the coast, but then eventually it turns around and it comes back, and so you end up with these huge retention zones within the surf zone. So what they did, is anybody familiar with his work? They put GPSs on people who wear wetsuits and flotation devices, and then they just go in, and then he must have the ambulance on call or something, and he uses these human drifters to see what's going on within the surf zone. Pretty cool. I'm not quite sure I want to sign up for that, maybe when there's not sharks and stuff around, but so there is some recirculation within the surf zone, which would just mean that if you're a larvae, you just washing machine for, yeah. And I think that's why, so I didn't show it today, but I also, during the coastal boundary layer work, looked at larval abundances within the coastal boundary layer. So people that have looked at larval abundances, especially up at Bodega, they pretty much stopped at that 30-meter isobath. So then I tried to fill in and see, okay, well, do we even see larvae within this coastal boundary layer region? And you do. There's a ton of larvae, but there's very few larvae right outside the surf zone. And so I would imagine that they don't want to be in that washing machine for whatever reason, or they've already settled. And so that's an interesting thing that I also want to follow up on. I think that's definitely this missing piece, especially, I think mostly about subtidal organisms, but when you think about what's going on in the intertidal, somehow they got to get on shore, and that, obviously the surf zone is involved in that. So I think the physics people are working on the surf zone, and hopefully with some of the data that Jamie's been collecting, we can start thinking about what that means for larvae.
So I'm going to ask you a question about your kelp force work.
Yeah.
From what I've heard so far, and what I've heard in seminars and stuff, the giant kelp itself, including the microscopic things, doesn't seem to be greatly affected. In fact, the spore plasmatics and the productivity would increase.
Yeah.
Right? So that suggests that most of the effects of changing in CO2 are going to be near the bottom. That's where most of the calcareous organisms are, etc.
Yeah.
So I wonder whether measuring at 1 meter off the bottom is really a useful place to measure.
Yeah. One of the things that we do want to do is to go out, and just like we're doing the high resolution with increasing what we're looking at through time, but we also want to do more resolution throughout the water column. And I think the 1 meter above the bottom thing just really comes from a logistical issue of the skin being open and all that kind of nonsense. But But yeah, I mean, there may be a pretty big amount of variability between right at the bottom and a meter above the bottom. But I'm not sure that, you know, that parcel of water will be that different.
Wouldn't there be a lot of drag and stuff down there? Or you get, what, you're running out
I don't know if it would be that, I mean, there's definitely like lower flows because you have the vertical boundary layer that's being set up there. But we do have days, I didn't show any of the, oh actually, so this is looking at, I'm just gonna skip ahead. Okay, so this is the CTD casts. So this is looking at vertical distributions of temperature as a function of time inside and outside the kelp forest. And this is, you know, going through the whole water column with the CTD casts. And so we get these days where it's really homogeneous all the way through the water column. And then we get these other days where, you know, we get pretty, you know, high amounts of stratification. Also other days where we get this like warm water lens that goes from the kelp forest and extends offshore, and then colder water offshore, maybe influenced by upwelling. And so, you know, starting with looking at these days when it's really homogeneous throughout the water column will give us a good idea of the contribution of kelp to the story and putting those measurements in context. But yes, it would be good to also look at right at the bottom.
And other groups do have sensors. I mean, at Hopkins, there are pH sensors that are down at the bottom. We also have one out at Stillwater Cove right now, too. It's,
Yeah.
it's, I mean, it's closest to the bottom. You know, it's probably this far off the bottom where the sensor sits. So,
Yeah.
so it's even closer.
There's also, I mean, every day when we go out and do these, I have moorings out there and we tie up to the moorings, but you know, some days the wind's going one way and so, you know, the boat hangs. So there's a little bit variability from day to day in what's going on with the tide. And sometimes, you know, there's a rock, and so maybe our, you know, CTD or our Niskin lands on a rock. So, you know, with all of that in mind, really we're just getting, you know, a characterization of that general environment, like, you know, 1 meter above the bottom.
All right, with that, let's thank Kerry, and then she'll be here.